As you're moving further, you'll find that we are talking about the developing nervous system at various levels. And you will see this diagram. It is the dorsum of the baby you are looking at. So when you look at the dorsum of the baby, there will be a neural plate formed, a neural groove formed, a neural tube formed. We want to talk about that. So looking at the dorsum of the baby, you will see that there's a neural plate, neural groove and neural tube, and then there is a dorsal view and there is a transverse section. This is a transverse section. First we will look at a transverse section, then draw the transverse section. Let's do that. See, if you remember, we have discussed earlier also, there will be a neural plate ectoderm and it will form a neural groove, and that neural groove will detach from the dorsal surface to become a neural tube, and that neural tube will form the central nervous system. Actually, this is a transverse section of neural tube. It's a long tube and it is going to form upper end, it is forming brain, and lower end, it is forming the spinal cord. So brain and spinal cord develop from neural tube, the CNS. If you're talking about the periphery, there will be some neural crest cells developing at the periphery, and they will also detach from the dorsal surface and they will contribute to the peripheral nervous system. Actually, neural crest cells, as they detach from the dorsal surface, they'll form most of the ganglia. Most of the ganglia in our body are derived from the neural crest cell, contributing to peripheral nervous system. Now, what about the most peripheral surface ectoderm? Actually, that most peripheral surface ectoderm, which is on the right side and left side, keep coming close in the midline. So as it is closing towards the midline, it will actually fuse in the midline. As the neural structure will detach from the dorsal surface, the surface ectoderm will fuse in the midline. As the neural structure will detach from the dorsal surface, the surface ectoderm will fuse in the midline to form the skin epithelium. So there will be surface ectoderm forming skin epithelium, which is skin epidermis, as the neural tissue has already come ventral. As you're talking about this development, you will find we are taking a transverse section of the embryo, showing the notochord again, which is the axial mesoderm. This notochord is important to stimulate the overlying ectoderm, if you remember. As it is stimulating the overlying ectoderm, that will become neural plate ectoderm. Now, if that is the neural plate ectoderm, then it is the one which is going to form central nervous system. In the periphery, we have some surface ectoderm. And what is that surface ectoderm doing? That surface ectoderm is going to form the skin epithelium. So in the periphery, maybe right side or left side, you have some surface ectoderm, and that will be forming the skin epithelium, skin epidermis. We told that there are some neural crest cells here contributing to peripheral nervous system. Peripheral nervous system. The neural plate has become neural groove. If you say that the neural plate has become the neural groove now, what was at the periphery of the neural groove? It was actually some neural crest cells, and those neural crest cells were contributing to peripheral nervous system. So this plate has become a groove, and that is the neural groove, and at the periphery you have some neural crest cells still there. And what is still more at the periphery? What is still more at the periphery, you can see, is this. What is this? That is the surface ectoderm. So as you are talking about the most peripheral structure here, what is the surface ectoderm? What is surface ectoderm supposed to do? Actually, when the neural tissue detaches from the surface, a gap will be created, and that gap will be filled by the surface ectoderm fusing in the midline. This neural groove will detach now to form the neural tube here. This is the neural tube. Now, if you say this is the neural tube which is formed now, if this is the neural tube which is formed now, it is a transverse section of neural tube. What has happened to the surface? Some gap was created. That gap will be filled by the fusion of the surface ectoderm in the midline. No more gap now. And this surface ectoderm was actually forming the skin epithelium once the neural tissue has detached from the dorsal surface. Some other structures also detach from the dorsal surface, which are the neural crest cells. So as the neural crest cells also have detached from the dorsal surface, because you see they are also on the dorsal side. As the neural crest cells also detach from the dorsal surface, they will be forming what? They form the peripheral nervous system. Let us discuss the peripheral nervous system formed by neural crest cell then. But before that, do you know what is this cavity here? Filled with some fluid called amniotic fluid. There you have amniotic cavity filled with amniotic fluid. So dorsal cavity is amniotic cavity which is filled with amniotic fluid. There will be some amniotic fluid on the neural plate. That amniotic fluid which is on the neural plate will be moving into the neural groove. And that amniotic fluid which is in the neural groove now will be inside the neural tube, and it will form the first CSF. Amniotic fluid will form the first CSF. Now, first cerebrospinal fluid is formed by amniotic fluid. Who will form it later? Later, the CSF is formed by choroid plexus once the ventricles are formed. So once the ventricles are formed, then the CSF will be contributed by choroid plexus, but the first CSF is contributed by amniotic fluid. Now this was a transverse section of the neural tube. Let us look at the neural tube in a complete view. This is the neural tube. Upper part forming brain, lower part forming the spinal cord, the CNS. So you are telling that this was a transverse section of neural tube, but the neural tube is a long tube, and in the upper part it will form the brain, and in the lower part it will form spinal cord. Yes, that is all coming from neural tube, forming the CNS. Then what about the ganglia? If you are talking about the ganglia, then they would have come from neural crest cell. Most of the ganglia come from neural crest cell, contributing to peripheral nervous system. So if you are talking about the ganglia, that is peripheral nervous system, and that would have come from the neural crest cells. Now once you have that kind of orientation, you are going to look at the ectodermal placodes also. In fact, what you saw was there was ectoderm, and there was this neural plate ectoderm, and as you said, this is the neural plate ectoderm. What was at the periphery? At the periphery there was some surface ectoderm. Now if you say at the periphery there is some surface ectoderm, fine. What is developing at the junction of the two? That was the neural crest cell. If you say that was the neural crest cell, fine. We have already told that neural crest cell is the fourth germ layer coming from epiblast, but there is one more thing to discuss, which is ectodermal placode. They will also develop at this junction and will be distributed among the surface ectoderm. So ectodermal placodes are also developing at the junction of the surface ectoderm and neural plate ectoderm, but are distributed in the surface ectoderm, and that ectodermal placode, of course coming from ectoderm, will be contributing to some neural tissue. As I told you, there are some ganglia which are not purely from neural crest cells. They are also from ectodermal placodes. So you mean to say that ectodermal placodes come from ectoderm? Yes, that is what is told by the books, and they are distributed in surface ectoderm for the initial period of development. Yes, and then they are contributing towards the ganglia. Yes, along with neural crest cell. Like which ganglia you are talking about? Maybe trigeminal ganglia, maybe vestibulocochlear ganglia. So trigeminal ganglia, vestibulocochlear ganglia, they are contributed not purely from the neural crest cell but also from the ectodermal placodes. Yes, that is the point here. Let us move on to the next thing. Actually, you were looking at the dorsal view of the baby, and a neural tube was formed. So upper part will form the brain and lower part will form the spinal cord. First, there will be a plate, but then plate will become a tube, and the upper part will form brain and lower part will form spinal cord. This is what we have to see. Now, you're looking at the dorsal view of the baby. Plate, groove, tube, and openings in the tube. Let us magnify the diagram. You are looking at a neural plate on the dorsum of the baby, back of the baby. There's a neural plate, but it is forming a neural groove. And after some time, there's a fusion in the cervical region. Do you remember we have discussed that which is the first pair of somite to form? It is in the cervical occipital region, day number 20. So day number 20, in the cervical occipital region, there is formation of some somite, the first pair of somite, and that is where the fusion of the neural tube is occurring. Neural tube formation begins in the cervical region, and then it will zip upward and zip downward. So as the fusion will zip up and zip down, then the full tube will form. As the neural tube is forming, you will find that still there are some openings. There is a anterior opening and posterior opening. Actually, we call this anterior neuropore in the brain side. See, brain will not form properly until the anterior neuropore is closed, and similarly the spinal cord will not form properly until the posterior neuropore is closed. They must close. See, anterior neuropore is closing by day number 25 and posterior is closing by day number 28, and if they don't close, there will be open neural tube defects. Open neural tube defects. There might be some anencephaly developing, or there might be some rachis developing. Now we want to talk about that rachis, and anencephaly also. When you talk about the neural tube defects, you have to understand that this is the baby, the head of the baby, and this is the body of the baby. But you're looking from the dorsal side. So this is head of the baby and that is the body of the baby. You're looking from the dorsal side, and what you found is there was a neural plate on the dorsum of the baby. Now this neural plate which was on the dorsum of the baby was actually having a fusion in the cervical region. And when there is a fusion in the cervical region, the initial fusion in the cervical region, neural tube formation begins, and now the neural tube has formed after some time. So as you say, the neural tube has formed after some time, you will also see that upper part of neural tube form brain and lower part will form spinal cord. It is all developing on the back of the baby. So upper part of neural tube will form the brain and lower part will form spinal cord, but there will be some openings still, and we are telling this is the anterior, or you can say the cranial neuropore. This is the anterior or the cranial neuropore, and then there is a posterior, that is a posterior or the caudal neuropore. They must close in due time. So as you say, this is the anterior or cranial neuropore, it must close by day number 25, and similarly the posterior neuropore, which should close after 3 days, that is day number 28. And what if the anterior neuropore is not closing day number 25, or if the posterior neuropore is not closing after 3 days, that is day number 28, then there will be open neural tube defects. So when you're talking about the open neural tube defects, you will find the mother is having high levels of alpha-fetoprotein. And when you found the high levels of alpha-fetoprotein, there is also high levels of acetylcholine esterase enzyme. Then you do the ultrasound and you found the problem. Problem number 1 could be anencephaly, or problem number 2, it could be the rachis. So as you see that the baby might be having a problem of anencephaly or rachis. If there is a non-fusion of the anterior neuropore, brain will not develop properly, leading to anencephaly. And if there's a non-fusion of the posterior neuropore, then it will be rachis. There will be an open spinal cord. So it is due to non-fusion of the posterior neuropore. Yes. Because there is non-fusion of the posterior neuropore, there can be rachis. Let us look at further details of the two babies. You find that in anencephaly, the brain is small and degenerated because it is exposed outside. But why it is exposed outside? And who is degenerating it? See, first of all, the brain is small and it is degenerated by amniotic fluid. But why it is degenerated by amniotic fluid? Because it is exposed to amniotic fluid. But why is it exposed to amniotic fluid? Because the skull cap is missing. Skull cap is missing. You mean to say there is some defect in the skull? Yeah, there is a defect in the skull. Skull is open, brain is open, and leading to degeneration of the brain due to exposure to the amniotic fluid. These babies, they don't even swallow the amniotic fluid because the swallow reflex is lost or absent. They don't drink amniotic fluid. That's why the mother will have more, which is called as polyhydramnios. So, swallow reflex is absent in anencephaly. That's why the baby is not drinking amniotic fluid. That is why mother has more, which is called as polyhydramnios. A lady with polyhydramnios came to you. What do? I check for alpha-fetoprotein. And what if you find it high? I check for acetylcholinesterase. And what if you find that high? Then I do ultrasound and I found anencephaly. Then I'll advise the parents for medical termination of pregnancy after taking consent. So you have to take the consent of the parent and advise them for medical termination of pregnancy, because such a baby with anencephaly is not desired to be born. It's not. You should not let it happen if possible. And what about the other baby, rachis? If you're talking about the rachis, that was a non-fusion of the posterior neuropore, and that is why there will be open spinal cord and CSF is leaking outside. So you are telling that CSF is leaking outside. Why? Because there is open spinal cord, and not only there is an open spinal cord, it is also exposed outside. See, rachis occurs because there is non-fusion of the posterior neuropore. And what will happen when there is a non-fusion of posterior neuropore? Open spinal cord. Not only there is a open spinal cord exposed, there is a CSF leaking outside. Now, if the CSF is leaking outside, you mean to say even the bone is open? Yes, there is an open vertebra. Actually, it is called as spina bifida. Skin is also open skin. And since the skin is open over the defect and vertebra is open and the spinal cord is open, that's why CSF is leaking outside in case of rachis. Now, we have to see some diagrams for them. And as you are talking about the diagrams, understand that you can prevent the neural tube defect. Prophylaxis of folic acid may be required. So you may be, it is perinatal. If you give 400 microgram per day of the folic acid, you are going to reduce the chances of neural tube defects. But it should be given perinatal. Start antenatal, give natal, and also postnatal. This is a baby with anencephaly. If you say this is a baby with anencephaly, why? Because there is a open, skull is open, skull is open, the brain is open, exposed to amniotic fluid also, because there was a non-closure of the anterior neuropore. What about the rachis? They're almost born dead. So there is a defect in the skin, defect in the vertebra, defect in the spinal cord also. And what is leaking outside? See, defect in the skin and vertebra, and what is leaking outside is CSF. CSF leaking outside, bacteria is leaking inside, killing the baby. See, can you understand rachis, why it happens? Rachis happens because the neural groove never became a neural tube. The neural groove never became a neural tube. It must detach from the dorsal surface. Then only the peripheral skin can cover the defect. Do you remember? there's a neural groove which will detach from the dorsal surface to form a neural tube. But if it does not detach from the dorsal surface, then the skin cannot cover it. Skin will remain at the periphery. And not only skin remains at periphery, but the vertebra is also open, spina bifida. Now, there is a condition called craniorachis. It is combination of the two things, which are, it is a combination where the entire neural tube is open, so it is anencephaly plus rachis. You see, this is a case of anencephaly plus rachis because the brain is open, skull is open, anencephaly. And the vertebra is open, skin is open, spinal cord is open, CSF is leaking outside. It is rachis also. So rachis plus anencephaly. Together it is called as craniorachis. You can see here that the skin is at the periphery and CSF is leaking outside. And there is open vertebra, spina bifida. So as you can understand that it is rachis. If that is rachis, then what is this? This is anencephaly. Together this is craniorachis. Now as you are looking at this question, which is one of the AIMS question, is it spina bifida occulta? What is occulta? Occulta means hidden. If it is hidden, then this is not hidden, this is manifesta. So you mean to say this is spina bifida manifesta? Yes, it is not occulta. Occulta means hidden. Is it hidden? Can you see it is hidden? It is such a big cyst there. It is spina bifida cystica. So if it is spina bifida cystica or spina bifida manifesta, it is already manifested. So it is not occulta. No. So is it spina bifida cystica with meningocele? No. Why? Because this is not a clear cyst. It has some lines, dark lines. So there are some dark lines and it is not a clear cyst. No. So it cannot be meningocele. No. Then what it is? It is spina bifida cystica with the meningomyelocele. That myelo name comes because some neural tissue has come here. So some neural tissue has come creating some lines and this is meningomyelocele. That is our answer. Yes. Why it cannot be craniorachis? Cannot be craniorachis because in rachis you don't see the covering of skin. And it is not a elevation, it is usually a depression. There's an open spinal cord, open skin, open vertebra. So this cannot be craniorachis. No. Why? Because it is not covered by skin. This anomaly is covered by skin. And okay, so shall I keep my answer as myelomeningocele? Yes, it is spina bifida cystica with meningomyelocele. Take a syringe, aspirate the cyst, and you will see that what you get is CSF in your sample. Now let us look at the details of spina bifida. The most common type of spina bifida is spina bifida occulta. What is occulta? Occulta means hidden. Hidden means it is hidden. You don't know it is existing. The most common type of spina bifida is spina bifida occulta. It is hidden. There will be just a tuft of hair on the back lumbosacral region. Nothing else. But if it is hidden, then how do you know that it is there? It is incidental finding. What is incidental finding? Means I was just taking some X-ray and I just found that the patient has some problem here and it is called spina bifida occulta. Otherwise it is hidden. Even the patient don't know. It is asymptomatic. Okay, what about the next one, which is spina bifida cystica with meningocele? If it is meningocele, then you see that some meninges have moved into the cyst on the lumbosacral region, and if you say so, it will be clear cyst. It is clear cyst. You can aspirate and you can get CSF out of that sample. So you are puncturing the cyst and aspirating CSF. Now it's a clear cyst. Yes, because only meninges have come out. And what in the next stage? Next could be that even the component of spinal cord has moved out. And if the component of spinal cord has moved out, then what will you call it as? It is called spina bifida cystica with meningomyelocele. So some of the neural tissue also have moved outside, so it is called meningomyelocele. This cyst will not be clear. It will have some lines, dark lines. And why do you have dark lines? Because of that neural tissue. See, meningomyeloceles in the lumbosacral regions, they are not clear. They have some dark lines, dark tissue is there. And what is that dark tissue? The neural tissue. Meningomyelocele, lumbosacral region. And what about the rachis-chysis? Rachis-chysis, we have already discussed that it is deadly. It is the least common to occur. And it is the most deadly. The babies are dead already. So there will be open vertebra, there is open skin, and the spinal cord is also open and CSF is leaking outside. As we are moving further, we want to talk about the developing brain and ventricles along with that. And in that process, you will see that a question has come asking, where is spinal cord developing from? Now, spinal cord, of course, spinal cord would develop from, yes, what is the answer? Spinal cord would develop from the neural tube, not the brain. See, this is all brain. What is mesencephalon? Midbrain. What is rhombencephalon? Hindbrain. What is the prosencephalon? That is the forebrain. So this is forebrain, and this is the midbrain, and this is the hindbrain. Yes. So you mean to say brain will not form the spinal cord? No. So that means the answer is neural tube. Yes, the answer is neural tube. That is the answer. Keep the answer and look at the details. One question is asking, third ventricle belongs to? Third ventricle is a midline ventricle. Okay, but belongs to? Third ventricle, which is the midline ventricle, belongs to the diencephalon part of the brain. Now, if you say diencephalon part of the brain, can you tell me what is in the telencephalon? In the telencephalon, I have lateral ventricles. Can you tell me what is there in the hindbrain? This is hindbrain. In the hindbrain, you have two components. One is called metencephalon and myelencephalon. What is the ventricle of hindbrain? That is the fourth ventricle. So you have got the fourth ventricle for the hindbrain, which will itself include metencephalon and the myelencephalon. But the answer remains diencephalon, because the question is asking third ventricle. Third ventricle. Answer is choice number B. Now, once you have this basic orientation, you will tell, in the beginning we have forebrain, midbrain, hindbrain, which is further dividing into 5 parts of the brain. Actually, in the beginning there is a single brain vesicle, 3 brain vesicle, 5 brain vesicle, and this is the telencephalon, then there's a diencephalon, then there's a mesencephalon, metencephalon, myelencephalon, spinal cord. All that we have to describe in detail. Okay, then let us do it. See, this is the 3 parts of the brain developing, which are called prosencephalon, the forebrain, mesencephalon, the midbrain, and the rhombencephalon, the hindbrain. Now, if you say it is like that, the forebrain itself divides into 2, which is called telencephalon and the diencephalon. Midbrain is mesencephalon, and it will not divide into any further components. It will remain as midbrain only. And what about the hindbrain, rhombencephalon? If you're talking about the hindbrain, rhombencephalon, it will divide into 2 components. One is called as metencephalon, which is pontocerebellum, and the myelencephalon, which is the medulla oblongata. Now, as you see, the telencephalon is having a ventricle system, which we told is lateral ventricle. Why they are called lateral ventricles in the telencephalon? Because they are going to the right side and left side. So as they are going to right side and left side laterally, they are called as the lateral ventricles. Lateral ventricles are the ventricles of the telencephalon, which is basically the cerebrum region. Telencephalon is the cerebrum region, and the cerebrum will have lateral ventricles on the right side and left side. What about the diencephalon, which is thalamus region? If you say diencephalon is the thalamus region, that is where we have the third ventricle. Actually, third ventricle is a midline ventricle, and it is the ventricle of the diencephalon. Then what is the ventricle of the midbrain. The ventricle of midbrain is also a midline ventricle, and it is called as the aqueduct of Sylvius. Now, the cerebral aqueduct of the Sylvius is a midline ventricle, ventricle of the midbrain. What is the ventricle of the hindbrain? Hindbrain has fourth ventricle, which is diamond-shaped ventricle, rhomboid-shaped ventricle. Now, when you say fourth ventricle is a diamond-shaped ventricle or rhomboid-shaped ventricle, which is the ventricle of the hindbrain, fine. What is the fourth ventricle continuing downward as? It is central canal inside the spinal cord. So, fourth ventricle will continue as the central canal inside the spinal cord then. Now, with that orientation, we proceed further. We are telling this is some choroid plexus in the lateral ventricle and also projecting into third ventricle. This is going to form the CSF, the ultrafiltrate of blood. So, most of the choroid plexus is projecting into the lateral ventricle. Some of that is projecting into the third ventricle and forming the CSF, cerebrospinal fluid. Though some choroid plexus is also present in the fourth ventricle and forming some component of CSF there as well. Mostly it is in the lateral ventricle. Also remember that when you are talking about the cranial nerve number 1, olfactory nerve, nerve of smell, it is a nerve of telencephalon, part of the forebrain. Diencephalon, part of the brain, will grow into the eyeball to become retina and the optic nerve. Optic nerve is the nerve of diencephalon. Nerve number 1 and nerve number 2 belong to forebrain. Now, when you say nerve number 1 and 2 belong to forebrain, then what about nerve number 3 to 12? Nerve number 3 to 12 come from the brainstem, and brainstem has 3 parts. What are the 3 parts of the brainstem? And they are giving cranial number 3 to 12. There is midbrain, which is followed by pons, which is followed by medulla oblongata. And brainstem will give you cranial number 3 to 12. As you are talking about the pontomedullary junction, there is a nerve developing in the brainstem. In the brainstem, there is a pontomedullary junction. A nerve is developing, which is the 8th nerve for hearing and balance. It is called vestibulocochlear nerve. Now, number 1 and 2 and 8 are pure sensory nerve. Now, number 1, the olfactory nerve. Now, number 2, the optic nerve. Now, number 8, vestibulocochlear nerve for hearing balance are pure sensory nerves. You have to understand that in the beginning it is a straight neural tube, but then it is folding to become C-shape brain. C-shape brain. So in the beginning maybe it is a straight neural tube, but later it will bend to become a C-shape. Why it will become C-shape? Because the brain is folding, cephalocaudal folding. You remember there was a cephalocaudal folding in the embryo? So there is a cephalocaudal folding and the structures are becoming C-shape. And you will see that this telencephalon was forming the cerebrum. This is cerebrum. The diencephalon, the yellow color, was forming thalamus. That is the diencephalon thalamus. This is midbrain mesencephalon. That is midbrain mesencephalon. That is the pontocerebellum, the metencephalon. This is pontocerebellum, the metencephalon. That is the myelencephalon medulla oblongata. This is myelencephalon medulla oblongata. Continuing inferiorly as the spinal cord. That is the spinal cord. Also, the ventricles become C-shape. Which ventricle becomes C-shape? The lateral ventricle. You see, this is the lateral ventricle which is becoming C-shape. The lateral ventricle will put the CSF into third ventricle, into fourth ventricle, into central canal of spinal cord. So, we have to discuss the developing ventricles now in a diagram. But at this moment, as you are talking about the parts of the brain, let us add some more details. As you see, there are 3 parts of the brain we have in the beginning. 3 brain vesicles which are called prosencephalon, mesencephalon and rhombencephalon. The forebrain itself divided into telencephalon, diencephalon. What do you include in telencephalon? Actually, telencephalon will be the cerebral hemispheres we already have told, but then we also have to include basal ganglia. And what do you include in basal ganglia? There are many nuclei, and some of them are amygdaloid nucleus, there is putamen nucleus, there is caudate nucleus. This is all basal ganglia, part of the telencephalon. In the telencephalon, you have learned that olfactory pathway is there. Olfactory nerve is the nerve of telencephalon. So, olfactory nerve is the nerve of telencephalon. Hippocampus, which is involved in recent memory, is also a part of the telencephalon, and it is also a part of limbic system. Limbic system, which is involved with the relation of emotions with the memory, is some Papez circuit. We will talk about the Papez circuit later. It is a part of the limbic system, but in Papez circuit you have to talk about emotions and memory. You have to talk about the hippocampus again. It's a part of telencephalon actually. Then what is in diencephalon? Diencephalon means the thalamus and related thalamus. What is related thalamus? It could be epithalamus, subthalamus, hypothalamus. Any thalamus is diencephalon. If you remember, we have told diencephalon part of the brain will have a downward extension to form the posterior pituitary, which is also called as neurohypophysis. And you also have discussed earlier that diencephalon part of the brain will grow into the eyeball to become retina and optic nerve. So it is the optic nerve, the nerve of diencephalon. Diencephalon was extending into the eyeball to become retina, optic nerve, and optic nerve is the nerve of diencephalon. And what about the pontocerebellum? If you say pontocerebellum, it is metencephalon, part of the brain. See, in the beginning, we have forebrain forming telencephalon, diencephalon, and the telencephalon of cerebral hemisphere, basal ganglia, which is shown here with the lateral ventricles. If you say forebrain, you have diencephalon, then it will include the thalami, which could be hypothalamus, epithalamus, subthalamus, even the retina and optic nerve, and it is having the third ventricle, which is the midline ventricle. Midline ventricle, third ventricle is the ventricle of diencephalon. What about the midbrain? Midbrain is not dividing any further. It is mesencephalon. It is having cerebral aqueduct of Sylvius. And what about the hindbrain? Hindbrain is two component. There is the metencephalon, which is pontocerebellum, and there is a myelencephalon, which is the medulla oblongata, and they have the fourth ventricle, which is a diamond-shaped ventricle, rhomboid-shaped ventricle. And what is the ventricle of the spinal cord? Ventricle of spinal cord is central canal. Let us draw these diagrams now. So as you're drawing these diagrams, you have to begin with neural tube. If this is the neural tube, you know, the neural tube is having circulation of some fluid. What is that fluid? That is amniotic fluid. And as the amniotic fluid is passing through the neural tube, it will form the first cerebrospinal fluid. But after some time, the anterior neuropore will close and posterior neuropore will close, so that you will have brain developing and the spinal cord developing. What will happen then? Will form the CSF then. Let us find out. This neural tube, the neural pores are going to close, and then you'll have what is called as the brain and the spinal cord. So this is the brain and the spinal cord now? Yes, because the anterior neuropore has closed, the brain has formed, and the posterior neuropore has closed and spinal cord has formed. Then where is the CSF? CSF is now trapped inside the ventricles. Now if you're telling the CSF is now trapped inside the ventricles, who is going to form CSF now? That time it is the choroid plexus. And what is choroid plexus? It is some capillary plexus. And what is that capillary plexus forming? The ultrafiltrate of blood. And what is that ultrafiltrate of blood? That ultrafiltrate of blood is nothing but CSF. So you mean to say that once the ports are closed and the CSF is trapped inside the ventricles, is the CNS hollow from inside? Yes, ventricles means hollow inside the CNS and filled with CSF, and that CSF is being secreted by ultrafiltrate of blood, choroid plexus, capillary plexus. Now let's go to the next part of the diagram. In the beginning, you have one brain vesicle, but later you'll have three brain vesicle. Let us talk about three brain vesicle now. In the beginning, we have one brain vesicle, but later we have three brain vesicle. What are the three brain vesicle you have got? I have got the forebrain, and then I have got the midbrain, and then there is the hindbrain. Now, if you are talking about the three brain vesicle, what are they further going to divide into? That we will find, but first we draw the diagram. This is forebrain, midbrain and hindbrain. If that is forebrain, midbrain, hindbrain, this must be a spinal cord. Yeah, that is spinal cord. This is spinal cord. Now, if you say that is a spinal cord, fine. Which ventricles do you have in the forebrain? I have ventricle 1, 2, 3. Ventricle 1, 2, 3 is present in the forebrain. Let us discuss the ventricle 1, 2, 3 in the forebrain then. See, they are arranged in a Y-shaped manner. This is ventricle 1 going to right side, ventricle 2 going to left side, and ventricle 3 in the midline. So you're telling that in the forebrain you have ventricle 1, 2, 3 and 1 and 2 are going right and left side? Yes, that is why they are also called as lateral ventricles together. So when you're talking about the lateral ventricles together, that is ventricle 1 and 2. Where is the ventricle 3? It is a midline ventricle and it is here. So they have some Y-shape arrangement, yes, in the forebrain. And what about the ventricle of the midbrain? If you say ventricle of midbrain, it is here and it is called as the cerebral aqueduct of Sylvius. Cerebral aqueduct of Sylvius. What about the ventricle of the hindbrain? It is actually fourth ventricle. And when you're talking about the fourth ventricle, the ventricle of the hindbrain, it is rhomboid shape. Let us draw it. This is rhomboid shape fourth ventricle. And what is the ventricle of the spinal cord? It is called central canal. That is the central canal. Central canal is the ventricle of the spinal cord. But before that, you have got the fourth ventricle. Now, CSF will be passing through these ventricles, but then it will escape also the ventricular system. How does CSF escape the ventricular system? You find that there is a midline opening and then there are two lateral openings also. What are you talking about? Actually, there's a midline opening which is the Magendie foramen, and there are two lateral openings which are called as the foramen of Luschka, Luschka foramina. There are two in number. So you're telling that there's a midline foramen which is Magendie foramen, and then remember MM, midline is Magendie, LL, the Luschka is lateral. These three foramina are present at the roof of the fourth ventricle. Now, if you say they are present at the roof of the fourth ventricle, these three foramina which are present at the roof of the fourth ventricle, what are they for? Actually, that is where the CSF is escaping into subarachnoid space. So you mean to say ventricles are leaking out the CSF into subarachnoid space by passing at the roof of the fourth ventricle through some foramina there? Yes. Why the CSF should escape here? Because the CSF is now supposed to be entering dural venous sinus. It has to enter dural venous sinus, absorbed by arachnoid granulations. Arachnoid granulations. Remember, if there is aqueductal stenosis, there will be internal hydrocephalus. What is this? Narrowing of aqueduct, aqueductal stenosis. So if there is some narrowing of aqueduct, which is aqueductal stenosis, there will be hydrocephalus. It is called internal hydrocephalus. Which ventricles will dilate? Ventricle number 1, 2, 3 will dilate. So, which brain will die? Due to flattening effect? And your answer is forebrain will die due to flattening effect from inside. See, in the beginning there is one brain vesicle, and then I have three brain vesicle, which is forebrain and midbrain, and then you have the hindbrain. Forebrain itself will divide into two. What is that? There will be the telencephalon, then there is the diencephalon. And what is the midbrain forming? It is forming the mesencephalon, and then there will be hindbrain which is dividing into two, which is called as the metaencephalon, and then there is myelencephalon. 5 brain vesicle. So in the adult brain, we will have 5 brain vesicle because forebrain will give telencephalon, diencephalon, the midbrain is the mesencephalon, and the hindbrain is the metaencephalon and the myelencephalon. As you say all that, you can put them in a diagrammatic form now. Let us draw the diagram. You first draw the midline of the body. That is the midline of the body. Now, once you tell this is the midline of the body, let us start with the forebrain. Let us draw the diagram for the forebrain. And there you show this is the forebrain. If you say this is the forebrain, which part of forebrain are you talking about? Actually, when you say cerebral hemisphere and the basal ganglia, so that is the telencephalon part of the brain. Then what is diencephalon? Diencephalon is here. And what is that? That is the thalamus, and not only thalamus but any related thalamus. So you have one thalamus on right side, you have one thalamus on the left side, and the thalamus region, or maybe epithalamus, metathalamus, subthalamus, whatever thalamus, is diencephalon, part of the forebrain. What about the ventricles? Ventricles, if you say, then it was ventricle 1, 2, 3. And can you show ventricle 1, 2, 3 now? Actually, ventricle 1 and 2 belong to telencephalon, and ventricle 3, the diencephalon. If you say so, draw the Y-shaped ventricles then. As you're drawing the Y-shaped ventricle, you will see this is ventricle number 1, ventricle number 2, ventricle number 3. So this is the forebrain having ventricle 1, 2, 3. Ventricle 1 and 2 belong to telencephalon. Ventricle 3 is the ventricle of diencephalon. Ventricle 1 and 2 are going lateral. One is going to right side, 2 is going to left side. They are laterally going, so they are also called lateral ventricles. Lateral ventricles are the ventricles of cerebral hemisphere and basal ganglia. Now, lateral ventricles are the ventricles of the cerebral hemisphere and basal ganglia. What about the third ventricle? Third ventricle is a midline ventricle. Ventricle of the diencephalon is sandwiched between the two thalami. And if you say the third ventricle is sandwiched between two thalami, you can also say thalamus is forming the lateral wall of the third ventricle. Now let us come to the midbrain. It will have a ventricle called aqueduct of Sylvius. Let us talk about that. First, you draw the midbrain. This is the midbrain. How do you know that this is the midbrain? How do you know? I know this is midbrain because I found some rounded elevation. What is this rounded elevation? There are two superior colliculi, there are two inferior colliculi. Now, if you say there are two rounded elevation superior colliculi, two rounded elevation inferior colliculi, where are they present? They are in the tectum part of the midbrain. Tectum part of midbrain, where? On the dorsum of the midbrain. So on the dorsum of midbrain you have tectum, part of midbrain. Yes. And on the dorsum of the midbrain, or the tectum of the midbrain, I have what is called as corpora quadrigemina. What is this corpora quadrigemina? Four bodies. Four bodies. Okay, these four bodies, yes. Actually, there's a midline ventricle here. What is the name of this midline ventricle? That name of midline ventricle is, of course, aqueduct of Sylvius. As you are moving on to hindbrain, you have to talk about metaencephalon and the myelencephalon. Let us draw them. As you are drawing them, you will find that this is the metaencephalon, myelencephalon. What do you include in meta and myelencephalon? Actually, metaencephalon, I have pons plus cerebellum. And what is myelencephalon? That is the medulla oblongata. Now, if you are telling that metaencephalon is pons plus cerebellum and myelencephalon is medulla oblongata, is this the medulla oblongata? Yes, that is the medulla oblongata. And if that is the medulla oblongata, then where is the pons? Pons will be, of course, here. Now, if this is pons, then where is the cerebellum? It is still developing. See, in the beginning, pons will develop, and later the cerebellum will develop. And pontocerebellum will be metaencephalon. Now, if you are including midbrain plus pons plus medulla oblongata together, this is brainstem. This is cerebellum developing now. If you say this is cerebellum developing, who is developing first? That is a pons, and later develop the cerebellum. This is the right cerebellar hemisphere, that is the left cerebellar hemisphere, and now the cerebellum is developing. Actually, pontocerebellum is the metaencephalon. Now, once you say that this is cerebellum, you know cerebellum attaches to brainstem with the help of three peduncles. This is superior cerebellar peduncle, this is the middle cerebellar peduncle, this is the inferior cerebellar peduncle. You mean to say this cerebellum is attaching to midbrain by superior cerebellar peduncle? Yes. And to the pons by middle cerebellar peduncle? Yes. And to the medulla oblongata by inferior cerebellar peduncle on the right side. Not only right side, left side also. You can show left side also. You tell this is the superior cerebellar peduncle, this is the middle cerebellar peduncle, this is inferior cerebellar peduncle. Actually, cerebellum attaches to brainstem. What is brainstem? The three parts of the brain. What is that? Midbrain by superior peduncle, pons by the middle peduncle, medulla oblongata by inferior peduncle. And then you have to understand, sandwich between cerebellum posteriorly and brainstem anteriorly is the fourth ventricle. Fourth ventricle is sandwiched between cerebellum posteriorly as the roof and brainstem anteriorly as the floor. Floor is brainstem, roof is cerebellum. Now, if you say cerebellum is the roof of the fourth ventricle, can I see the fourth ventricle? The ventricle of the hindbrain. If you say ventricle of the hindbrain, which is the fourth ventricle, yes, we will see that it is a rhomboid-shaped ventricle. But to discuss the fourth ventricle, ventricle of the hindbrain, rhomboid-shaped ventricle, first you do one thing: put a midpoint of medulla oblongata, upper half of medulla oblongata and lower half of medulla oblongata. Why do you do that? For we are putting a line here telling this upper half of medulla oblongata, this is lower half of medulla oblongata, because we understand that the fourth ventricle will come only till the upper half of medulla oblongata. Now, if you say fourth ventricle is a rhomboid-shaped ventricle, diamond-shaped ventricle, and coming till the upper half of the medulla oblongata, then what is there in the lower half of the medulla oblongata? That is where the central canal will begin. And this central canal, which begins in the lower half of the medulla oblongata, will continue into the spinal cord. Also, it will continue in the spinal cord. Actually, you can show the spinal cord there like this. This is spinal cord. So that is a spinal cord? Yes. You are telling that fourth ventricle is a rhomboid-shaped ventricle and it is present only in the upper half of the medulla oblongata. Yes, because in the lower half of medulla oblongata you will find there is a central canal which will then extend into spinal cord as the central canal only. Now, what I am trying to tell is this fourth ventricle is having a roof of cerebellum posteriorly and having a floor of the brainstem anteriorly, especially the pons and upper half of medulla oblongata. So what is there at the floor of the fourth ventricle? It is the pons and upper half of medulla oblongata. This you remember. It is sandwiched between brainstem anterior and cerebellum posterior. Now this is what we wanted to know here, and we can proceed further. Then we are talking about the sagittal view of a developing brain. When you take a sagittal section of developing brain, you will find it is actually anterior-posterior section. Now, when you are taking anterior-posterior section of the brain, remove one half of the brain and look at the other half of the brain. This is going to be a medial view of the one half of the brain after taking anterior-posterior section. And we are looking at developing brain. So it is here. I told you the brain will become C-shape. Then what is the axis on which it is becoming C-shape? The brain is going to become C-shape on an axis which is actually the thalamus. Thalamus becomes the axis, on that the brain is becoming C-shape. This is the diagram. And see, this is the thalamus which is the axis. So you have to remember that the first commissure to develop is anterior commissure. What is commissure? Commissure fibers. You know, commissure fibers are axons connecting right brain with the left brain. And if you're connecting right brain with the left brain, there are some commissure fibers. They are crossing axon, the midline, and this is anterior commissure, connecting brain anteriorly, and it is the first commissure to develop. Now, if you say first commissure to develop is anterior commissure, then which is the next one to develop? It is the corpus callosum. And as you say, corpus callosum is also commissural fiber. You mean to say they are also axons? Yes, they are axons connecting right brain with the left brain, crossing the midline. This will be corpus callosum. The next commissure to develop is going to become C shape. In the next diagram, you can make out that. So let us magnify the second portion of the diagram, and as you are magnifying, you can see for yourself that the corpus callosum is going to become C shape. Not only that corpus callosum, but also not only corpus callosum but also fornix. What is fornix? Fornix is also collection of axons. If it is also collection of axons, it is becoming C shape. Then what is it connecting? Actually, it is output tract of the hippocampus, part of the telencephalon. You remember hippocampus is a part of telencephalon. It will send one outgoing fiber, and that is the fornix. So fornix is the efferent tract. Yes, it is actually major efferent tract of the hippocampus. Where the hippocampus is found in the adult brain is a question. Hippocampus in the adult brain is present in the temporal lobe, inferior lobe. So there is inferior lobe or temporal lobe, that is where you will have the hippocampus located. And then you are looking at the caudate nucleus, part of the basal ganglia. If you say that is the caudate nucleus, part of the basal ganglia, is it also going to become C shape? Yes, it is. Let us see the C shape of caudate. Caudate means comma shape. It will become comma shape, caudate, comma shape, C shape. And it is evident in the third part of the diagram. As you look at the third part of the diagram, you will find this is the caudate nucleus, which has become C shape. Actually, it will have anteriorly present head and the tail. And then what is this? This is the C shape cerebrum. Cerebrum also becomes C shape. Developing some lobes. What are the lobes of cerebrum developing? Let us find out about them also. And before you do that, you draw the diagram. So what is the axis? Axis is the thalamus. If you say thalamus is the axis, then what is the C shape holding it like this? That was actually caudate nucleus. So caudate nucleus is a C-shaped nucleus holding thalamus like that. The head was anterior and the tail was anterior. Now, if you say this is the sagittal section of the brain and you are telling thalamus is the axis on which various structures are becoming C-shaped, this is the caudate nucleus, part of the basal ganglia. Yes, it is the caudate nucleus and it is the part of the basal ganglia. Where is hippocampus present? Actually, hippocampus is present in the telencephalon part of the brain. Yeah, telencephalon, but where? In the temporal lobe. So it was in the temporal lobe, telencephalon part of the brain. Fine. Hippocampus. What is hippocampus for? Hippocampus, it is the site for the short-term memory. And if I want to turn the short-term memory into long-term memory, yeah, that is a very big issue. You know, we all have short-term memory, but we are unable to convert it into long-term memory. That is a problem with everybody. But you have to learn. How can you convert the short-term memory into long-term memory? Because hippocampus has short-term memory. It requires an output tract. What is that output tract? That is a fornix, which is becoming C-shape. Oh, this is the fornix. Yes. And this fornix is becoming C-shape. Yes. And it is a major output tract from the hippocampus. Yes. And this is the one which is going to help me consolidation of memory. Yes, it is the one which help you in consolidation of memory. And you will find if it is lesion, there will be amnesia syndromes. Amnesia syndromes. Have you seen the movie Gajni? Amir Khan? This was the lesion. Sir, what are you talking about? It was a fictional character. Yeah, fictional character can be logical. So you mean to say in Gajni, Amir Khan was used to write down everywhere because his short-term memory was not becoming long-term memory? Something wrong with fornix? It's a remake of English movie, you know that? So hippocampus, short-term memory, if it wants to consolidate memory, then should send the information via fornix, which is the major output tract, and if it is damaged, then consolidation memory is compromised. There can be amnesia syndrome. By the way, where is this fornix C-shaped structure carrying the information towards? It's a part of Pepper's circuit. Yeah, okay, part of Pepper's circuit, I understand. Pepper's circuit, which itself is a part of the limbic system. And this limbic system, which is involved with some memory and also the emotions, understood. We have discussed this earlier, isn't it? That Pepper's circuit is something to do with limbic system, is something to do with memory and emotions. But point is, when hippocampus is sending information via the fornix, where is the information going? It is a circuit. It must complete. It is sending the information towards the hypothalamus, which will then send it to the thalamus. And then the thalamus will send it to the cerebrum. Which part of cerebrum it will send the information? See, the hippocampus is sending information towards the hypothalamus, is sending it to the thalamus, is sending it to the cerebrum, because once the memory is spread across the cerebrum, it becomes long-term memory. So once the memory is spread across the cerebrum, then it becomes long-term memory. But anyway, this is not a pathway for consolidation of memory only. It is also a pathway which is a circuit. You must complete the circuit. So to complete the circuit, I come back to hippocampus and circuit is complete. You mean to say cerebrum itself will have some communication with hippocampus then? Yes, the circuit is complete. Let us see a diagram for the same purpose. You'll find here a diagram for Pepper's circuit, limbic system. See, this is the hippocampus, and what is it sending? The C-shaped structure, that is a fornix. Where is the fornix going? To the mammillary body. Where is mammillary body? Hypothalamus, and from the hypothalamus to the thalamus. See, hippocampus sending via fornix into mammillary body. Mammillary body is hypothalamus, and from there it will go to the thalamus. Which nucleus of thalamus? That is anterior nucleus of thalamus. And from the anterior nucleus of thalamus, where is it going? To the cerebrum. Which part of cerebrum it is going? To the cingulate gyrus. Cerebrum, cingulate gyrus. And from there, where is it going? Where? It is a circuit, it has to complete. It will come back to the hippocampus. Where is cingulate gyrus? Here, cingulate gyrus. This is cingulate gyrus. So like that, you just remember some component of limbic system for memory and emotions. Now let us move on further and discuss some more details of cerebrum and ventricles, because we have to look at the CSF circulation also. You see, this is the C-shaped cerebrum now. And as the cerebrum is becoming C-shaped, it will develop some lobes. Which lobes it is developing? The lobes which the cerebrum is developing is on the anterior side, frontal lobe, on the posterior side, occipital lobe, inferiorly the temporal lobe, and there is also a parietal lobe. So once the cerebrum is becoming C-shaped, it will develop some lobes and some sulci also. Sulci we will discuss later. At this moment, can you see the lateral ventricle, which is ventricle of cerebrum, is also becoming C-shape, and it will develop a frontal pole, there will be a occipital pole, there will be a temporal pole. Now most of the CSF is formed in the lateral ventricle, will be poured into the third ventricle, which is the midline ventricle, and from the third ventricle the CSF will be poured into the aqueduct of Sylvius, and from the aqueduct of Sylvius will go into the fourth ventricle. If you remember, the fourth ventricle is sandwiched between the brainstem anteriorly, chiefly the pons, and cerebellum posteriorly as the roof. So cerebellum was posteriorly as the roof of the fourth ventricle, and the fourth ventricle is actually sandwiched between brainstem anteriorly and cerebellum posteriorly. This is what we have discussed earlier as well. Now, as you are talking about the ventricles, understand there is the C-shaped ventricle, which is lateral ventricle, and it is now having the C-shaped ventricle is now having some poles. What are the poles? You see, it is inside the cerebrum, so accordingly it will have one anterior or the frontal horn of lateral ventricle, posterior or the occipital horn of lateral ventricle, inferior or the temporal horn of lateral ventricle. Actually, lateral ventricles are the largest of the ventricles in the largest part of the brain, which is cerebrum, forming largest amount of CSF and poured into the third ventricle, which is the midline ventricle, through this opening, which is called interventricular foramen of Monro. Foramen of Monro. So you are having some interventricular foramen of Monro, and where is that sending the CSF from third ventricle? No, it is actually lateral ventricle sending the CSF into third ventricle by interventricular foramen of Monro. And where the third ventricle will send the CSF into the fourth ventricle, passing cerebral aqueduct of Sylvius. So after passing cerebral aqueduct of Sylvius, the CSF will come into fourth ventricle, and it can escape the fourth ventricle via some openings on the roof. Could be Magendie foramen, or could be the Luschka foramen, or it may happen that some CSF continue into central canal, into the spinal cord inferiorly. That is the CSF circulation, but we need more information. And this is the front view of the brain. If you look at the front view of the brain, again you can discuss that lateral ventricles are having C shape. This is the lateral ventricle on one side, this lateral ventricle on the other side. They are pouring the CSF into the midline ventricle, which is the third ventricle, by some interventricular foramen of Monro. Then the third ventricle is sending the CSF into the fourth ventricle, into the central canal. If you take a coronal section of the brain, look from the front view, you will find that these are the lateral ventricles, and they are sending the CSF into the third ventricle, midline ventricle, into the fourth ventricle, and into the central canal. Fourth ventricle is a V-shaped ventricle or a rhomboid-shaped ventricle there. We have drawn this diagram already. Do you remember? But we need more information. Actually, you have to look at a sagittal section now and discuss the CSF circulation in more detail. And what you do is, now look at this diagram. You have to understand that CSF, which was in the lateral ventricle, will be passing through foramen of Monro to enter this third ventricle, which is a midline ventricle, and then enter into the aqueduct of Sylvius to enter the fourth ventricle, and then it will continue into the central canal. It may also happen that it is escaping out through the various opening, which could be Magendie or Luschka foramen, so that it will come into this space, which is called as the subarachnoid space, and from the subarachnoid space it can move into what is called as arachnoid granulations, and from the arachnoid granulation it will be absorbed into the dural venous sinus, like this is the superior sagittal sinus. So you are telling about the circulation of the CSF here. Yes, let's draw a diagram for that. And as you're drawing the diagram, you will tell there were lateral ventricles. Now, if these are the lateral ventricles and they are forming the CSF, where is the CSF coming from there? Actually, there is one opening. What is that opening? That is called as the interventricular foramen of Monro. And that Monro foramen, what is it doing? It will let the CSF escape from the this lateral ventricle into the third ventricle. And when it will come into the third ventricle, when the CSF will come into the third ventricle, it will be going towards what is called as the cerebral aqueduct of Sylvius. So when it is going towards the cerebral aqueduct of the Sylvius, then where is it going after that? So lateral ventricles was having CSF formed, passing interventricular foramen of Monro, coming to the third ventricle, and after third ventricle it is going to cerebral aqueduct of Sylvius to enter the fourth ventricle. And when you are talking about the fourth ventricle, you'll find, what is it sandwiched between? It is sandwiched between a structure which is posterior, which is called as the cerebellum, as the roof of the fourth ventricle, whereas the floor, you have the pons, and not only pons but also upper half of medulla oblongata. So you are telling that fourth ventricle is sandwiched in such way that cerebellum is posteriorly as the roof, and then you have the pons and the upper half of medulla oblongata as the floor of the fourth ventricle. Yes, that's correct. And then it will be continuing into the central canal of the spinal cord. It will continue into the central canal of the spinal cord. But then some CSF can escape the roof of the fourth ventricle, and as we have known that there can be opening in the midline, which is called as the Magendie foramen, or there can be lateral openings which are called as the Luschka foramina. So there could be a midline Magendie, there could be a lateral Luschka foramina. What are they for? Actually, as you are following the CSF, you see this CSF has come and it was there in the fourth ventricle. It is escaping at the roof of the fourth ventricle through the foramen of Magendie to enter what is called as subarachnoid space, and in the subarachnoid space it will continue, and then it will be actually absorbed by arachnoid granulations. As it is absorbed by the arachnoid granulations, where is it going? From there it is going to enter what is called as dural venous sinus. If you say that is dural venous sinus, what is that name here? It is superior sagittal sinus. So ultimately the CSF will be absorbed by arachnoid granulation into dural venous sinus, which is superior sagittal sinus. It is ultrafiltrate of blood. It will go back into the blood, venous blood. So CSF is ultrafiltrate of blood and will go back into the blood, which is the venous blood, and travel towards the heart. All the venous blood will travel back toward the heart, come back into the arteries. Arteries will form the capillaries. Capillaries will form the choroid plexus, and the choroid plexus, capillary plexus will form ultrafiltrate of blood CSF in the lateral ventricle. So circulation is over. You know that the heart will set the arteries, will have the capillaries, the choroid plexus will form the ultrafiltrate of blood CSF in the lateral ventricle, which is circulating inside the ventricle, but also it has to be absorbed back into the blood. And which blood? That is the venous blood, that is the dural venous sinus, which will then drain towards the heart. Venous blood drains towards the heart. What about the developing brain from the front view? You have taken a coronal section now. So you have taken a coronal section of the brain and remove the anterior part of the brain and look at the front view of the brain. You want to look at the developing brain from the front view, coronal section. And when you do that, you will find this is the diagram. Choroid plexus, capillary plexus, which is projecting into lateral ventricle. And not only it is projecting into lateral ventricle, but it is also projecting into the third ventricle, which is the midline ventricle. This is the midline ventricle, the third ventricle. Do you remember third ventricle is the midline ventricle is sandwiched between the two thalami? That is where the choroid plexus is projecting to form CSF. Now this third ventricle, which is sandwiched between the two thalami, not only it is sandwiched between the two thalami, actually thalami will form the lateral wall of third ventricle. Now when you say thalamus will form the lateral wall of third ventricle, what is under the thalamus? It is called hypothalamus. And hypothalamus, which is under the thalamus, is forming the floor of the third ventricle. Hypothalamus, which is under the thalamus, forming floor of the third ventricle, is not only floor, it will also contribute to lateral wall. So it is contributing lateral wall and the floor of the third ventricle. That is the hypothalamus. What is this here? This is the corpus striatum, part of basal ganglia. Now if you say this is corpus striatum, part of the basal ganglia, you find that there are some axons bisecting it. Some of the axons are bisecting it. They are called as projection fibers. They are projecting from the higher brain center to the lower brain center, and from the lower brain center to the higher brain center, and later they will form the internal capsule. What is the internal capsule? What is a capsule? Collection of axons. Capsule is collection of axons. So internal capsule is some projection fiber connecting the upper brain with the lower brain and vice versa, is bisecting corpus striatum into two parts. So if it is bisecting corpus striatum into two parts, what is more lateral? More lateral will be lentiform and more medial will be the caudate nucleus. Let us look at a diagram which will tell you about the details, the next part of the diagram. See, this is the next part of the diagram where you are showing some projection fibers, some collection of axons which are connecting upper brain center with the lower brain center and vice versa. Internal capsule is some projection fiber connecting upper and lower brain center is bisecting the corpus stratum of basal ganglia in two parts. And remember, lentiform is always lateral to the internal capsule and caudate is medial to internal capsule. This will help you in orientation of the adult brain also. So these are two parts of the corpus stratum. And the lentiform is lateral to internal capsule and the caudate is medial to internal capsule. And what about the third ventricle? See, lateral is lentiform nucleus, medial is caudate nucleus, and if you are talking about the third ventricle, it is the midline ventricle, sandwich between. It is a midline ventricle which is sandwiched between the two thalami, and not only the two thalami but also the hypothalami. Hypothalami which are sandwiching third ventricle in the midline are also the floor of the third ventricle. As you say so, I have to draw a diagram for that. Let us draw the diagram then. And as you are drawing the diagram, you will tell that this is the Y-shaped ventricle. If you remember, there was a Y-shaped ventricle. Where? In the forebrain. Ventricle number 1, ventricle number 2, ventricle number 3. 3 is a midline ventricle. Ventricle 1 and 2 are also called as lateral ventricles because they are going to right and left side. Now, once you have shown the three ventricles here, the third is the midline ventricle, you also tell what is this structure which is forming the lateral wall and the floor. Actually, first you have thalamus forming the lateral wall on each side, and then what is under that? Under that is hypothalamus on each side, which is not only forming the lateral wall but also the floor. So third ventricle is a midline ventricle and you have thalamus on each side and forming the lateral wall of the third ventricle. But not only thalamus, it is sandwiched between hypothalamus. Hypothalamus is not only lateral wall but also floor. At the floor of third ventricle, I will tell you some structures. Just remember the names. At the floor of the third ventricle, they are part of the hypothalamus. The names are, number 1, tuber cinerarium. Comes as a question. And number 2, it is mammillary body, part of hypothalamus. So we're telling there are two structures which are at the floor of the third ventricle which are part of the hypothalamus. Yes, one is called tuber cinerarium and one is called mammillary body, part of hypothalamus. Now, once you have that kind of orientation, fine. We are talking about the corpus stratum. What is happening to corpus stratum? Actually, corpus stratum, which is a part of the basal ganglia, was bisected by some axons running up and down. And those axons which were running up and down are actually called projection fibers because they are projecting from upper brain center to lower brain center. Later, they will form the internal capsule. And this internal capsule was actually bisecting the corpus stratum into two parts such that the lentiform nucleus is always lateral. And what is medial? That is the caudate nucleus. So caudate nucleus is medial and lentiform is lateral to the internal capsule. This orientation we wanted to have.